AAV, and in the later half of the talk, I'm going to talk about our recent work on SARS coronavirus 2. So first, the improved muscle targeting AAV. So AAV is not the virus we have been working in our lab, but what the graduate student who just helped me with this computer change wanted to work on gene therapy vector. And because my husband, the lab, Michael Farzan, use AAV to express the antibody against the HIV, I thought it was a good idea to do so. And so we specifically focused on improving AAV on muscle targeting. So because we lost a lot of time, I'm going to skip some slides. So why we want to target skeletal muscle? Because intramuscular injection is clinically very convenient, and we human beings have lots of AAV antibodies against many different serotypes. So this pre-existing antibody is a very big problem for the efficiency of AAV gene therapy vector. But if you inject through intramuscular, then you can avoid contacting some of these antibodies. And skeletal muscle lives for a long time. And if you are producing therapeutic protein and you want it in your circulation, that muscle is a good place to produce this protein because it has very rich vascular supply. So it's a good place to produce a therapeutic protein to diffuse it into circulation. So that's a intramuscular injection. And then AAV. AAV has advantage as a gene therapy vector. It is one of the most sought after viral gene therapy vector because it's non-pathogenic and it has a low immunogenicity. And it does not integrate, but not zero. So it integrates once in a while, but with the extremely low frequency. And it has a history of success. Luxturna and Zolgensma were approved by FDA in the US, and Factor 9 was approved only in Europe. Luxturna is the treatment for genetic eye blindness, and Zolgensma is to treat muscle atrophy for children under age 2. And this doesn't happen all the time, but some monkeys given intramuscular injection with the AAV1 expressing one of the HIV neutralizing antibody still express, after longer than a decade later, still express therapeutic level of antibody, but this doesn't happen all the time. AAV, I talked about this AAV, and AAV is non-enveloped virus and icosahedral, so it does not have a lipid membrane. And it's a DNA virus, and it has only 2 genes, Rab and Cap. But it depends on helper viruses like adenovirus or herpes virus, or helper proteins. You can provide as a proteins instead of a whole adenovirus or herpes virus. So it has only 2 genes. Rab gene does not encode polymerase, so AAV depends on cellular polymerase. So it produces 4 genes. The first 2 genes are involved in replication, and second protein, and second 2 proteins are involved in encapsulation. And cap gene produces VP123 from the same frame, and then another assembly activating protein, which is not specific, which is not required for some AAV serotypes. I will talk about this VP later, so I'm going to go on. So, okay, so AAV is one of the best viral gene therapy vector, but there are still challenges. What are the challenges? First, the high cost of production is prohibitively high. So Luxturna cost $1 million to treat both the eyes. And the titer, purified vector titer we can get, considering the small injection volume, is sometimes prohibitively high. So if you convert the vector titer necessary for rhesus macaque to express therapeutic level of protein, then human adult of 90 kilogram will require 1.3 times 10 to the 14 vector genome in 1 or 2 ml. So this is very high. And more importantly, the anti-drug antibody. So that's the antibody we generate against the therapeutic protein. This is a big problem because once these antibodies are produced, then therapeutic protein level in serum goes way down and it becomes useless. And the thing is, AAV capsid protein and DNA genome work as adjuvant. So if we can reduce the amount of AAV capsid and DNA genome by 10 times, then probably the anti-drug antibody production is reduced also. So basically, if we increase efficiency, lower dose, that lowers cost, and also lowers immunogenicity, and it will in turn then increase apparent vector efficiency. Back again. So which AAV and how are we going to do it? So there are many more AAVs, but these AAV 1 through 9 are the most frequently used. And what's listed here are the attachment factors. They used to be called the AAV receptors, but now the protein receptor for all AAVs is identified that we should call this attachment factors. So which AAV are we going to use? We chose AAV9 simply because in mice, AAV9 was shown to be the most muscle tropic vector. So in past decades, people used many different methods to increase the transduction efficiency of AAV or change tissue tropism. So this kind of method, so we are going to use a different one. That is, we wanted to target a receptor highly expressed on differentiated muscle. And how do we do that? We using a ligand that binds the receptor with a high affinity. And we chose this receptor, the IR, insulin receptor, as the target receptor. Why? We initially thought that other muscle-specific receptors, but we realized that these muscle-specific receptors are expressed mostly in neuromuscular junction. And we do not want to interfere with the neuromuscular junction even temporarily. And we don't know at neuromuscular junction, these receptors are exposed. So then we learned that IR receptor is highly expressed at terminally differentiated muscle. And because IR being the famous receptor, so a lot of study was done. So in the literature, there was an insulin receptor, insulin mimetic already selected from phage display. And its affinity to insulin is not, insulin receptor is not as high as that of human insulin, but nonetheless, it is very high. So we inserted this 36 amino acid peptide into the variable loop for N8 of AAV9. So I said that this capgene expresses 3 different viral protein, 1 to 3, from the same frame. So they share, all 3 of them share C-terminal 2/3 of the protein. And BP1 has a phospholipase A2 and nuclear localization signal. BPT has a nuclear localization signal, but all of them have a same C-terminal 2/3, and C-terminal part is the exposed part to outside. And this phospholipase A2 and nuclear localization signal are tucked inside the virus. So, and all these 3 BPs have 9 variable loops, 1 through 9. And because they are undistinguishable, that so if you insert something at variable loop 4, then it's going to be in all capsid proteins. And so another reason we chose loop 4 and 8 is because those are already shown to be amenable for manipulation. And more importantly, loop 4 and loop 8 are located in this is threefold axis of symmetry that is most protruding on the surface of AAV. So as you see here, loop 4 is hanging in the air to outside, and loop 8 also. So we inserted S519 peptide at the tip of loop 4 or loop 8 and made the virus. The first experiment was so disappointing that we almost ended this project. Even if the transduction efficiency is increased, if your vector yield is reduced significantly, it's no use because the cost goes up. The yield was reduced, and at the same vector genome, the infectivity is even more reduced. So then before we stopped this project, we gave another thought. That is, maybe we are wrong that even though this loop 4 and loop 8 seem to be hanging in the air, that if you put 36 amino acids, the peptide, they are going to be expressed all like this and they wouldn't have any problem. But maybe this is 36 amino acids, the peptide is folded this direction. So they have a steric hindrance, they have a conflict with each other. So if we make them to have only 2 S519 peptide or 1 peptide per 34 axis, that maybe that works. So before we gave up, we tried it. So we mixed, in addition to helper plasmid and transgene cassette, in this case expressing firefly luciferase or GFP, we mixed this wild type and mutant capsid plasmid at various ratio. Cody Jackson, who helped with my computer, is the one who led this project. So he tested various ratios, and at 100% of mutant capsid, as you can see, the yield was dropped by tenfold. And then at the same vector genome copy, he tested infectivity and learned that at 10:1 ratio of a wild type to S519 inserted capsid, the infectivity is the highest. So we decided to call that EAAV9, that enhanced the AAV9. This is the case for loop 8 insertion, and we did the same with the loop 4 insertion. And that was tested in 293 T cells because all cells express insulin receptor. So we made insulin receptor knockout cells, and these control cells are the 293 T cells that went through the same process of selection as IR knockout cells, except that guide RNA, instead of guide RNA, the scrambled guide RNA was used. And as you see here, wild type AAV9 barely infected either cell. And with the loop 4 S519 infectivity transduction increased, loop 8 S519 transduction increased. Lassa was a control, and Lassa doesn't care insulin receptor, so that infected both the cells equally. So this is the quantification of this type of experiment. So both the 4 EAV9, 4 and 8 showed significant increase in transduction in the presence of IR, insulin receptor. So next, we wanted to see this effect in human skeletal muscle cell. So we purchased the human myocytes looking like that and put it in differentiation media. After several days, these cells fused to form myotubes like this. So Cody used these myotubes to do a transduction efficient transduction assay using wild type AAV9 or EAAV9 in the presence or absence of IRFC, that is a extracellular domain insulin receptor fused to FC domain, used as an inhibitor here to show that this entry transduction is insulin receptor dependent. So wild type AAV9 barely transduced these cells, and EAV9 dramatically better, and IRFC inhibited that transduction, and autofluorescence is shown to show cell density. And interestingly, this EAV9 was very specific for myotubes. So obvious next experiment is to mouse experiment. And before we go directly to mouse, of course, we had to test the mouse insulin receptor, because up to this point, we used only human insulin receptor. Mouse and human insulin receptor genes have a very high homology. So we knew that it was going to work, but nonetheless, we had to test this wild type AAV9. So we got mouse brown pre-adipocyte cell line that express insulin receptor and insulin-like growth factor receptor. So both of them are knocked out. That's a double knockout. And in that double knockout cell line, mouse insulin receptor was reconstituted. That's a blue cell. So, and we saw that EAAV9 used the mouse insulin receptor to transduce this murin IR reconstituted double knockout cells. So we are ready to go to mouse. So we made wild type and EAAV9 vectors expressing firefly luciferase and injected into gastrocnemius muscle, that's a leg muscle. And 2 weeks later, there were images, then you already see that EAAV9 is more efficient. And Cody measured this at week 2, 3, and 4. And as you see here, although there is a big variation among the mice in the same group, nonetheless, there is a significant increase in transduction efficiency by EAAV9. Right, that's one thing. But the other thing is that by this time, Cody has done many of similar experiments. But what he noticed was that somehow in the first mouse experiment, he got a lot bigger difference between wild type and EAAV9. And that effect somehow become smaller and smaller. And he started wondering what in the world happened, what changed, because he thought that he was doing the same thing all the time. And then he got to this point, because we are targeting insulin receptor, and that's going to be affected by insulin and glucose, because insulin works as an inhibitor because it has a higher affinity to IR, insulin receptor, than S519 peptide. And then he realized that AAV injection, he used to do AAV injection at a certain time, and that has been changing little by little. So as many people know, which I didn't know, that mice eat during the night. So in the morning, in normal fed mice, glucose level is high. So to work on this high glucose level, insulin level goes up during the day. But as you see, if mice are fasted, then insulin level is low. So of course, when we inject IR targeting AAV, we want insulin level low because they work as a competitor. So Cody did, we withdrew food early in the morning, and 4 hours later, injected mice with AAV9 and EAAV9, and further fasted for 4 more hours while AAV transduction went on, and then returned the food. and image then, yes, you can see the difference between wild type EAAV9 is much bigger. And so, yeah, now they have nearly, this is a log 2 scale, nearly 20-fold increase in mouse muscle. And I can just skip this, but let me see, time. Yeah, I can just skip this. It just shows that although IR is expressed throughout the body, if you inject this vector through IV, the effect is not that great. So this vector is more suitable for IM injection. When we say that we are targeting IR receptor, then people are freaked because how do you manipulate the insulin receptor? What about glucose level? So we tested the glucose level using insulin as a control. So when insulin, so this is measurement before any injection. So when you give insulin, then glucose level is dropped, and to compensate it, then glucose level shoots up. So this is normal. And compared to that, wild type AAV shown in green, the variability is so big, but it does not change much, except that with the time, glucose level goes down a little bit because these mice are fasted. And EAAV9 level is not very much different from that of wild type. So although we already knew that the amount of vector we are using and number of S519 peptide, and it has a lower affinity to IR compared to insulin and all that, we knew that it wasn't going to do anything about systemic glucose level, but here is the proof that it doesn't. So then, because this is just a peptide inserted into loop, that we decided that this should work in other AAV serotypes. So we put this S519 into the variable loop 8 of AAV1, 2, and 8, and saw that some modest improvement in transduction efficiency or significant improvement of transduction efficiency. And interestingly, it improves the transduction efficiency of NP22. NP22 was engineered and selected in Mark Kay's lab for muscle tropism. So as you see here, it is already good for targeting muscle, and S519 peptide increased it even further. So this is a portable, transferable technique. And oh yeah, I almost forgot. Before the mouse experiment, of course, we did a cell culture experiment to see if this is worth the mouse experiment, because mouse experiment to save mice. As you see here, AAV1 in IR knockout cell, and this is a IR positive regular to control 293T. It's increased, but it is modest, 3, 4, 5-fold, or no change at all in vitro. So we almost didn't do a mouse experiment, but then Cody insists that we should, and he has a rationale, and that was a correct rationale, and he's a smart guy. So that his rationale is that the reason we don't see this difference by EAAV is because attachment factors for these viruses are abundantly expressed in 293T, and that might be different in skeletal muscle, and that's exactly right. So that's what happened. So especially for AAV, in vivo data and in vitro data, you cannot extrapolate in vivo result from in vitro result, actually. So the conclusion, because I ate up a lot of time, I'm going to just skip conclusion because it's a repeat of what I already said, and I will skip acknowledgement also. So I'm going to talk about SARS coronavirus, the mechanism of this D614G mutation. Okay, this D614G mutation was detected for the first time in January 2020. That was longer than a year ago. So why do we talk about it? Because it's an old story, but not because every single variant, current variant, has D614G mutation. When you look up, for example, UK variant and look up the mutations they carry, G614G is not mentioned at all, because now they consider this the G614 as wild type. So that's not even counted as a mutation. But so what I'm saying is G614G is not an old story. It's in every variant of every isolate of SARS coronavirus 2. I'm not going to give an introduction of SARS coronavirus 2 because I think that you guys by now heard so many talk on, listen to so many talks on SARS coronavirus 2, but this I have to talk about. So SARS coronavirus 2, like SARS coronavirus. So for convenience, I'm going to just say SARS-1 and SARS-2. SARS-1 used these two entry mechanisms. So does SARS-2. So one mechanism is that, as Mike introduced, we identified the ACE2. We, and in collaboration with Mike Farzan's lab, identified ACE2 as the receptor for first SARS-1. And it's good that SARS-2 also uses ACE2, I think it speeded up the speed of research of SARS-2 very much. Yes, I just want to take credit for that. And so another very important molecule is TMPRSS2. It's expressed on the cell surface. And if TMPRSS2 cleaves S protein on the cell surface, then virus fuses with the plasma membrane. So that is the entry location. But if TMPRSS2 is not there, or TMPRSS2 somehow does not cleave S protein, then through ACE2 endocytosis, it's internalized to late endosome or lysosome, and S protein is cleaved by cathepsins, cathepsin L, the most importantly, and fuse in the endosome. So these two pathways, and this is an S protein, and forget about all these small domains, but the important thing is that first the SARS has two domains, S1 and S2, but it was not cleaved here at the junction. But SARS-2, SARS-2 is cleaved by furin in the producer cell. That is the important part. SARS, in the case of first SARS, it's cleaved by TMPRSS2 or cathepsin in the target cell. But in the case of SARS-2, it's cleaved by furin. And there is another cleavage site, essential cleavage site is upstream of a fusion peptide that is called the S2 prime site. And both for SARS-1 and SARS-2 have to be cleaved by either TMPRSS2 or cathepsins in the lysosome. And the mutation we are talking about, D614G, is located toward the C terminus of a S1 domain. In the case of SARS-2, because it's cleaved, we should call this S1 and S2, S1 subunit and S2 subunit instead of domain. And I just want to point out that this RBD is located at the C-terminal half of the S1 subunit. And this residue 614 is in this green area at the C-terminus of S1, and that is in association with the S2. So I'm going to skip this, but Betty Corbert's group first detected this D614G mutation, and after March last year, it really predominated fast the world. And so we made a murine leukemia virus, pseudovirus, coated with D614 or G614 S protein, and tested their infectivity in 2930 expressing human ACE2 or human ACE2 plus human TMPRSS2. And as you see, in both the cells equally G614 pseudovirus infected a lot more efficiently. And we did a similar experiment in NCI HE1975, but long epithelial cells. And this cell did not express ACE2, so we had to provide the ACE2 by transduction. But as long as there was ACE2, the G614, higher infectivity of G614 is observed. For this D614G, as long as it is infectivity, everybody agreed that there was absolutely no problem. The problem came when the mechanism was studied. So there were roughly 3 major mechanisms proposed. One is enhanced cleavage at S1 and S2 junction and S2 prime site. This hypothesis did not get much support. There are few publications, but they started talking about S2 prime site because in the case of SARS-2, furin already cleaved at this junction. But nobody ever showed any increased cleavage at S2 prime site or even increased the cleavage at this site, at junction. So this is not really proved or supported by data. Second one is that increases spike with one-up RBD conformation. So it was shown even with the SARS-1 already that when you make a spike trimer, it was easy in the case of a SARS-1 because furin that did not cleave the junction of S1 and S2. So no S1 is dissociated. They saw that there are 2 populations, one RBD up population and all 3 RBD down population. This one up population, one RBD up, is better, is more suitable for ACE2 receptor binding. So there are these 2 populations, and what they are saying is that with the D614G mutation, this one up population portion is increased. That's what they are saying. Okay, so the third hypothesis is our hypothesis. It's not our hypothesis, but we proved it with our data that S protein stability is increased. There was a hypothesis that S protein stability is decreased, but anyway, and so that reduces the S1 shedding, and that increases, as a consequence, it increases functional spike protein. If there is only S2 on the virion, that's not functional. So S1 is still there, then that's a functional spike. So increase functional spike proteins on the virion. So I'm going to talk about our work first and come back to that one, second hypothesis. So we started with the murine leukemia virus, PV, and purified it through 20% sucrose cushion and ultracentrifugation. And as S protein, we use the N-terminal and C-terminal double flag tag for the purpose of detection because we didn't have any other means to detect. And so the purified viruses are directly run on the gel and Western blotted with the flag tag antibody. And as you see here, D614G, S1 and S2 band, and G614, S1 and S2. The total S protein is a lot more with the G614 virus. And S1 and S2, S1 to S2 ratio is higher with the G614 virus also. The quantity of a virus is controlled by P30, amount of P30, that is MLB GAG protein. So there is a significant difference in total S protein in D and G. And to see the S1, S2 ratio, let me check the time. Oops, I'm going to skip it because it is pretty late, so I'm going to skip it. So we then repeated the same experiment with the VLP, that's virus-like particle. Virus-like particles are made only with the SARS-2 proteins. So envelope protein, membrane protein, nucleocapsid protein. So it's more like a virus. So again, we had to use the NNC double flag test protein, but we basically got same result. So there are total more spike on G, and S1 and S2 ratio is higher with G also. And in this case, controlled amount of virus is controlled by N protein. And S protein is detected by human, oh no, I'm sorry, it is an flag tag. So it was detected by flag tag antibody. So, so then because other people did not see this difference, that we had to prove that it is a difference between D and G spike density and S1 shedding is not caused by flag tag, placing flag tag at the NNC terminus. So, and by then we had this plasma derived from convalescent patients, so we could detect S1 and S2. By the way, even though I ate up a lot of my time, that I have to make one point. A lot of COVID patient serum do not recognize S1. Most of them recognize S2 very well, but not S1. This already tells us that this SARS-2 virus does not have much S1 on their surface. That is, I'm talking about the original virus. Anyway, so in this case, S protein is untapped, and we detected with this convalescent plasma that recognized both S1 and S2. And we basically got, and this is a purified MLB PV, basically got same result. So why didn't other researchers see the spike density difference or higher S1, reduced S1 shedding with the G? And they were all published in a high-profile journal. So as a matter of fact, a few groups reported increased stability, but they were all published in not very noticeable journal. And then the others, some people analyzed the S protein in the cell lysate. That doesn't make any sense, but there was only one or two. And other people, I mean, in the papers published in Cell and Science and Nature, they had the all cytotoxic problem. Yes, so what I'm trying to say here is that we are the only correct people and other people are all wrong. That's what I'm saying, but that is correct. This cytotoxicity is so deeply associated with all this PV system and live viruses. Lentiviral PV, VSV PV, they are very, very toxic. By the time you guys, if you guys use this system, you guys would know. By the time you harvest that, maybe 48 hours, VSV PV transfected cells are all dead and lentivirus are half dead. Live virus, not to mention, they kill cells like crazy. VLP, the same. Why VLP? The most toxic protein to make VLP is M protein. So how did we see the result with the VLP? Because we had to adjust the M protein ratio very much to reduce the minimum amount of M protein. And we harvested the VLP. Now, we didn't wait until 48 hours. We harvested earlier to minimize this cytotoxic effect. So why does cytotoxicity mask the difference? Because S protein in the cell from the dead cells are released and they contaminate purified virion. We saw it ourselves, so we can say it with confidence. So we repeated our experiment with the lentivirus, that cytotoxic system. But if you harvest the PV earlier and optimize the condition, you can see the difference between D614 and G614, even with the lentiviral system. Anyway, so what about this one of RBD conformation? So many people in high-profile journal reported that one of population is increased in G614 trimer. The problem is, to make a trimer, all these people either mutate furin site or introduce a stabilizing diproline in the S2 domain. And so their experimental designed not to see stability difference. That's one thing. And the other thing is that one up many people observed the one up conformation is increased, but the increased degree of increase is not good. One paper talked about twice the increase, but other people talk about 30% increase or so. But even so, no matter what, so if one up conformation population is increased, then their affinity to ACE2 should be increased also. But actually, they never talk about ACE2 affinity, but one paper measured it, and actually that was decreased. So it just doesn't make sense. And one thing is that one Cell paper published that not only one up population is increased, but they saw 2 up and 3 up in G614, while all other papers or other structure biologists never ever saw 2 up or 3 up population. So 2 up and 3 up populations are total artifact, and they are in the way to disintegrating, probably. While these things went on, there showed up a paper from Bing Chen's lab at Harvard, deposited in bioRxiv, but now it's in press in Science. And this figure, first figure, tells us everything. That is, so he, instead of making this soluble trimer, he transfected the cells with a full-length G614 or G614S protein, and using mild detergent, he solubilized the membrane and column purified the trimer portion. With the G614, he saw at least three different peaks. With the G614, he saw only one peak. So peak 1 is a pre-fusion trimer, meaning they have a stable S1 and S2. Second peak is a post-fusion trimer that has only S2. And those peak-looking things in between must have one S1 or two S1. And peak 3 is shed S1. So this already shows that G614 has a lot more functional spike trimer functional spike trimer, and it does not shed S1 as much as G614. And in conclusion, they said the enhanced infectivity of the G614 virus largely results from the increased stability of the S trimer rather than the better exposed RBD. Why he said rather than better exposed RBD, you can read his paper in Science soon yourself. So because one picture is better than 1,000 words, we try to do cryo-EM. I'm going to skip this because if you use a full-length S protein, you barely see any spike, even though I can see the spike in G614 tendency of a higher spike on G614 PV, but nonetheless, it is very, very hard to see spike. So meanwhile, what we did was that we truncated the cytoplasmic tail of S protein, because this cytoplasmic truncation makes a spike trimer a lot more incorporated into the virion. And that is true for MLB, that is true for VSV and lentivirus all. We saw this with the first SARS. Anyway, this shows the Western blot of, So this DCT, the point is that even though a lot more spike is incorporated into the virion, the G614, the stability of phenotype is still maintained. So when you infect full-length S protein pseudovirus or DCT pseudovirus, this is D and this is G, so G is still a lot more efficient in infection. There is about 1 log difference. And before we send it to cryo-EM, we saw the spike density on Western blot. And as you see, for full length, you cannot even see spike S1 and S2 band. And delta CT incorporates a lot more S protein. But between D and G, the phenotype is still maintained. So we sent this to University of Washington, and they did cryo-EM. And what we learned is that it's not like all G614 virus, pseudovirus have less spike and all G614 have more spike. No, it's not like that. It's very various. This G614 virion has a full spike around it. This has a low to medium, and this is bold actually. And the G614 has a higher tendency to have a full spike. So as you see here, there are full spike and low, medium and low. So we counted many virions and came up with, of course, this is not real quantification, but this is just a pseudo quantification. They are at University of Washington, they are doing tomography. So that's going to give us a real quantification. But anyway, so we categorized these virions into 4 categories: high spike density, medium, low, and bold. And as you see, the original Wuhan D614 tended to have a low spike density in red, and G614 mutation has tended to have a high spike density on the virion. So this shows that, okay, yeah, we are right, that S1 shedding is reduced and spike density in the virion is increased by this mutation. So why this mutation is acquired by the virus? That is to compensate the instability resulted from the S1, S2 junction cleavage. That's what we believe. And we don't have time, so we can discuss this, some outstanding questions in the next session. I can take questions if you are still there after a long hour.
Thank you, Hyeryun. That was,
Hello.
and sorry that you had so many technical challenges at the beginning. So we do.
Let me see.
Oh.
Hello.
Yes, yeah, we're all here.
I cannot hear you. I think you need to unmute yourself.
I'm not muted.
Oh, it's me. Okay. Could you repeat it? Yeah.
No worries, no worries. So we have a question in the chat, and then I'll follow up with so the AAV virions, you made AAV virions whose capsid was a mixture of wild type and then engineered spikes from different ratios.
Mm-hmm.
How is that possible? How did you make that?
With that, you just mix, when you transfect, you just mix 2 plasmid, wild type plasmid, and the plasmid that contains mutation.
And then they just show up at the same ratio as the transfection?
Well, that we don't know. So what we know is the transfection ratio. So what I showed is transfection ratio. Actually, it's a good question. We do not know. It might be, mutant might be a little lower in terms of expression. Who knows? But that's a transfection ratio. Yes.
Yeah. So
they might have different. They might have different stabilities too.
That too. Yes. Yes. Right. So in real ratio, we do not know. Yeah.
And I also noted that you have these in your last cryo-EM images, mm-hmm. the mutant form of spike was also present at higher density, mm-hmm. which is really interesting. So do you think that it doesn't have it may not have necessarily a higher affinity for the ACE2 receptor, but because of the density changes, maybe it's able to enter the cells more efficiently?
Actually, this D614G mutation does not change affinity to ACE2 at all. So yes, the higher spike density was the mechanism. That was what I have been saying. Yes. Yes.
And then, so that's an interesting way by which viruses can be more efficient at entering cells.
Right. If you look at the cryo-EM of first SARS-1, because it's not cleaved by furin, the spike density is very high in that virus. But this SARS-2, because it's cleaved by furin and lose S1, if you go through all the cryo-EM of a virus, the paper, published the paper, it's pathetic. Spike density is really pathetic. There are only countable number of spikes, but with this mutation, the density is increased. So restored in a sense, actually.
Right. Yeah, yes. Yeah. That's so fascinating because, yeah, it just shows that you don't have to increase per se the affinity
to the
itself.
receptor.
You just can increase the density, and then it's an indirect way of making the process more efficient.
Yes, absolutely. In the case of first SARS, there is no room to increase spike density because it's completely
pretty high.
covered, right? Yeah, but in the case of SARS-2, that worked really. Yes.
Any other questions from the audience? I feel like I'm kind of hogging the little bit of time that we have available.
Ilham, there's one in the chat.
I already said that, Bert.
Oh.
Well, I mean, that was, I very much enjoyed the first part as well. And I never thought about AAV, if you're transfecting a gene that's important for a function, you have to think about the physiological process that you're manipulating, and the timing of the day of what you're trying to do could actually have a huge impact on the efficiency of the process.
Isn't that interesting? It's only because we chose the insulin receptor, but maybe other molecules are affected by circadian rhythm.
Oh, yeah. There's tons of molecules that are affected by the circadian rhythm. And I mean, the clock exists in almost every organ system. So that's just something to think about that I really never really paid that much attention to, but that was really compelling.
Yeah.
How much of an impact it had. Well, I apologize again for how many, the slow start at the beginning, but thank you for sticking it out with us and, working through those challenges.
No, it is my computer problem. So I apologize that I ate up in so many minutes, and I thank everybody who remained and listened to this talk. Thank you very much.
It was really a nice talk, Hyeryun. I really, really enjoyed it. It's probably, the computers probably also had it with the pandemic.
Infected with the mutant virus.
Exactly. The computer is done with all the Zoom meetings. So I think you're meeting with the students now. So I will, and Nick,